right. Kia ora koutou katoa. Ko Mick Black toku ingoa. Nā mai, haere mai. Welcome along to our Friday afternoon Genomics Aotearoa seminar series. The numbers keep increasing. This is incredible. We might hit 100 before I stop talking. Who knows? If it were Peter, we'd hit 200 before he stopped talking. That's why we muted him. Right. Enough frivolity. Peter promises to bring the frivolity at the end of the talk rather than the beginning. So I will stick with just introducing our speaker for today. We are lucky enough to have with us today Dan Tompkins, the science lead at Predator Free 2050. Dan is going to be talking to us today about genetic pest control. And I'm going to leave it at that, Dan, because I actually lost the blurb for your seminar. It's behind all the people and I can't find it again. So I'm going to hand over to you and tell us all about it.
That's good enough. That's all people need to know. Okay, I'll just get the slides going.
I will say what you're doing. I will say, just say thank you very much for coming along. I've been wanting to hear this talk for a while, so I'm going to sit back and enjoy it.
Yeah, all good. Well, thanks, Mick. Let me just start that. How's that looking?
Yep, looks good.
All good. Fantastic. Tēnā koutou katoa, ko mam tō te munga, ko waita awa, ko Ngāti Ingarangi te iwi, ko Otipoti te kanga, ko Dan Tompkins tokengoa. Nora, tēnā koutou, tēnā koutou, tēnā koutou katoa. Thanks for joining me on this Friday afternoon. I'm based down here in Dunedin as well with Peter, and so we do bump into each other every now and then. But it's my privilege really to have been working on New Zealand's Predator Free 2050 conservation mission for the last 8 years now, almost 9 years. And so it's been an interesting journey so far. We've made some really good progress, both on the ground in terms of getting elimination done, and also in the tools and tech space. And one part that I'll talk about today that I've not really talked about publicly, going into some of the details, is our funding and support of genetic pest control guidance and exploration for Predator Free 2050. And that's the topic of today's talk. Very briefly, you may already know all these details, but this is New Zealand's mission to eradicate invasive mustelids, possums, and rats from the whole of New Zealand by 2050. So we're talking about 27 million hectares of land area across not only backcountry and conservation estate, but also urban and rural landscapes. But we have some confidence that we can do this. Before the mission was launched, New Zealand had shown that we can eliminate these pests off offshore islands, and we maintain some good mainland sanctuaries with good biodiversity, and the predators kept out. And so this is really stepping up. Can we aim, or we're working to achieve, the entire elimination from the entire country of the mustelids, the possums, and the rats by 2050? We're now at 2025. We've got over 100,000 hectares of land where we've actually managed to eliminate predators from. We're now in a phase where we are filling in the remaining capability gaps before the strategy is to accelerate from 2030 onwards and to really start rolling out. Upon mission launch, or even before mission launch, this is back in 2012, Sir Paul Callaghan was one of the early people behind Predator Free 2050 in its current iteration. We've spoken about Predator Free New Zealand for a lot longer than that. But Sir Paul saw Predator Free as an Apollo program for New Zealand. The fact that we don't yet have all of the tools and the technologies and the capabilities to get this job done. And so a focus on innovation and science is needed alongside the public support and the feet on the ground to give us the capability of getting the national elimination eradication job done. And so why are we exploring genetic pest control tools? Well, as Sir Paul said, we need new tools and technology, and you just have to look at landscapes like this from the west coast of the South Island. You get a good idea why. Much of our country is in very hard to work landscapes. We also have other issues in urban and rural landscapes where we can't use the tools that are so effective on the conservation estate. And so we do need new tools and technologies to complement the current toolbox that we have. The second main reason, there's several, but just the two big ones. One is this inaccessibility, large, hard to work landscapes. And the second is rats. We're pretty good at controlling and eliminating possums and mustelids, Their life histories are such that they have relatively slow life histories, relatively large home ranges, that actually eliminating, controlling every individual isn't unfeasible and can be done. When it comes to rats, that's a whole different story. I mean, so we have small home ranges, we have rapid life histories. If you're in a city and there's a rat living in a compost bin, if you don't control that compost bin, you can't eliminate rats. And so rats are the one where we can eliminate them now, but the cost of doing so with our current range of tools would be prohibitive. And so we need new technologies, new tools to come alongside our current toolbox to help bring that cost down to something that we can afford to do as a country. And this is where genetic tools comes into part of the picture. We need new technologies to help us. And genetics is one area of several where there is that potential to give us new ways of doing things that could be a lot more cost effective than our current approaches. And so for the rest of the talk today, I'll just go through 5 bullets really. I'll talk about some of the modeling we've been supporting, some of the social and cultural considerations behind genetic pest control tool development and application, very briefly touch on international policy engagement we've been involved in, and then getting into some of the technical work, some of the preliminary explorations that we've been funding, and then where the technical development has got to date. One thing I won't talk about today, just because of the time limit, is there's a lot of genomics work going on in the background as well, with people like Alana Tatago and Steve Truick's group out of Massey and people at Lancair Research. There's a lot of supporting genomics happening, which I'm not going to go into today because that would need its own talk, its own half an hour. So quickly, some of the first work we funded, of course, was the mathematical modeling. And for this, we pulled in one of the leading groups in the world from Cornell University. This is Philip Messer's group. And over 2 to 3 years, his group really produced a slew of papers modeling the potential effectiveness of genetic control tool approaches. This is for rodent populations. And the information, the data, the parameters, that information was provided by Lancair Research, so that we had a leading international modeling group, but they were using the correct information and insights from rat populations in New Zealand. We also supported Philip and his group to produce what's called Slimer3, which is their modeling software, which is free to use. And so it's quite a good tool for others to start to play around and explore modeling pest control dynamics as well. Later on, we also, through a capability funding round, supported Anna Clark out of Otago. She's doing a modeling PhD on genetic pest control of rodents in multi-species communities. And Anna's been over to the States and done some work with Philip and co. And she's also done lots of other bits and pieces on the side. She seems to get into everything, whether it's research transparency, whether it's selecting genes to use for genetic control. There's quite a slew of interests there, but the focal piece there is actually looking at the multi-species modeling in a spatial landscape. You've probably all seen many results like this, and at the very high level of all the modeling work gone on for genetic pest control, not just for mammals, but for insects as well. It's really like, sometimes it works, sometimes it doesn't work. Sometimes it sort of works if you get the right parameters and the right characteristics or the right release strategy. And this is from Philip's Slim3 model, where you had the introduction in red of the individuals carrying a fertility suppression haplotype. And this is one of the fun occasions where actually resistance doesn't really evolve, but you get these predator-prey chase dynamics where you don't quite get elimination, but you do get control of a sort. This is just one of the types of control that have been simulated at certain parameter levels. Other types of control, you get effective elimination predicted by the models. And the real utility of these models is that it tells you initially which approaches theoretically can have the control, the suppression, or the elimination effect that you're looking for. And for those ones, what characteristics in the development do you need to aim for to actually realize that potential? Second piece of work I'll talk about today and that we've supported is social considerations. And so this is work that was started in the Bilateral Heritage National Science Challenge. And this is work led by Edie MacDonald, who was with the Department of Conservation at the time. And there's a couple of papers out now. I should mention that all the papers that I'm showing here I know I've missed off the journal details, but if you go to our website, we've got a resource library and you can find all of these resources on our website. So a couple of early papers there from Edie and Co, good multi-institutional publication, exploring public perceptions towards new technologies in general for pest control and gene drive specifically. In more recent years, social explorations have been picked up by Ocean Mercer and her group at Wellington. This work funded through Genomics Aotearoa. We've also supported a lady called Ali Palmer, who's been based out of Auckland, and she's been also one of our capability development investments, this time for her postdoc. And she's produced this report that's available on our website about the social and ethical challenges to Predator-free 2050. So this report goes a lot broader than genetic tools, but it has got some bits on genetics. It's a good read. Overall, though, with these social considerations, I haven't got the time to go into the detail, but the general messaging is that there is generally moderate levels of support for the development and the research into using genetic tools for Predator-free 2050. Of course, there's concerns from several parties about both the technical and the social feasibility of actually using these kinds of tools in the environment. And this is a concern that's reflected internationally as well. And I'll move on to that in a minute. But to me, though, the key finding that came out from all of the work, both Edie's and Ali's, is most people are asking for more information, more knowledge, more understanding. Even for scientists, genetics can be quite confusing. For non-scientists, it can be a complete black box. And so even now today, there's still a big piece to do on better empowering informed decision-making in the general public and also policymakers. I mean, they're also people as well. And we've got to actually work better on that education and communication and helping people to actually understand what all this is about. Moving on to cultural considerations as well. Again, back to Ocean Mercer and her group at Vic Uni. Again, this is work that was started under the Biological Heritage National Science Challenge, and then co-funded alongside that was funding from the company going through Genomics Aotearoa. And this is a piece that's come out quite recently at the end of 2024, looking at the Maori view of Has No Act. Just again, very briefly, some of the conclusions coming out of this is that first, future legislative review should give effect to Te Tiriti, whakapapa and Maori considerations in regulation. We should have cultural impact assessments based on both whakapapa and Maori, and also we need Mataranga-based environmental impact assessments alongside the traditional impact assessments that get conducted. This is the first piece of published work coming out of this stream, but I think there's quite a lot more to come, so watch this space. Moving into the international policy sector, this is something that I've been quite keen on, both keeping abreast of and informing, because this is a new topic being developed and debated quite hotly sometimes around the world. And so we've been involved in both IUCN and UN CBD processes. Most of the work that we've done has been through the IUCN. And I think the most tangible outcomes to date from it has been working on a policy task force for the IUCN, where one of the first steps that we made, this was 2019, I was producing a technical assessment of the current state of genetic frontiers for conservation, considering both synthetic biology application for the purpose of conservation and other impacts of synthetic biology for other purposes on biodiversity. A lot of the thinking that went into this still holds today, and actually it recognizes that there is a balance of potential positive and negative effects of using GM and synthetic biology in the environment. And the conclusion from it really, and this is the conclusion that's being carried through now into motions for the IUCN's policy on synthetic biology, is that really we need case-by-case risk assessments. We need wide consultation, appropriate consultation, case-by-case risk assessments, and a precautionary principle that favors conservation outcomes. Again, this is something else that could take a whole half an hour to talk on, But where we are at the moment is that the piece I was involved with developed a motion for the World Conservation Congress in 21, which was passed with over 80% of the 1,000+ member organizations voting in favor. And that was just to establish that the IUCN would develop its own policy on synthetic biology in relation to nature. And then the actual policy itself has been developed by a different task force more recently. It's going to the vote in October at the next World Conservation Congress. It's a very good balanced piece of work. It's had wide consultation. Again, it's basically saying we need case-by-case risk assessments, we need wide and appropriate consultation, and we need to favor conservation outcomes. It's currently the topic of online debating right now. If you're interested in that kind of thing, that'll be happening through to the middle of June. You can go to the IUCN website if you want to get involved in that. Moving into the technical part of the talk now, and some of the preliminary explorations. And so you're all probably aware that genetic control development for invertebrates is a lot further developed than it is for mammals. Peter himself on this talk is leading an MB with Amanda Black on seeing can we develop some of these control approaches for wasps. And overseas, there's been quite a few successes to date with things like mosquitoes and fruit flies. With mammals, it's a different story. We've seen good progress overseas with mice in the lab, which I'll touch on in a minute. But in terms of using genetic control tools or gene drive specifically even for mammal pest control, it's still very early days for most species. And for most species, there's been a lot of talk about using these kinds of tools, but the groundwork is still to be done. In this theme, we've been supporting a few bits and pieces of these exploration work. One of the critical hurdles, if we wanted to use genetic tools for possum control in New Zealand, is of course, as you may already know, is that gene editing is so much harder for marsupials than it is for eutherian mammals because of the issues with the hard shell around the egg. And so doing manipulations that can actually take in the embryo and be inherited is extremely difficult to do. It's been shown for the opossum over in the Americas. We funded Tim Horn, Mel Laird, and we put some co-funding alongside an MB Smart Ideas that they had to see could they actually develop methods for gene editing the brush-tailed possum. And unfortunately, it's still not possible. There is overseas work ongoing for other marsupials, and so we're going to watch that space very carefully. On the mustelid side, being a eutherian mammal, it should be a lot easier to use gene editing to develop these kinds of tools for. But really, our understanding of gene function for any mammal that's not mice and maybe rats is a lot lower. And so we're still in the phase of trying to work out what would be target genes for mustelid genetic population control. And for this piece of work, we funded Andrew Veal at Manaki Whenua Landcare Research, again co-funding this time alongside a Marsden Fast Start grant. Have a think about would, for example, the prospect of manipulating embryonic diapause in mustelids be one way to actually reduce their breeding. So you may or may not know female stoats can carry fertilized embryos at a paused state for months, waiting for the right environmental conditions before they then continue their development. If we can understand the genes and the genetics behind that diapause, can we actually use it to suppress breeding? There's some work being done by Andrew, as I said, potential mechanisms identified, but that exploration is still in the early stages as well. So moving on then to what we actually have been doing and supporting for rodents. The rodent development internationally for genetic control is much more developed. The rodents being lab animals are always going to be, mice in particular, are always going to be the place to try and develop these mechanisms first. If there's any mammal that's most like an insect, it's a mouse. And our understanding of them is such. So back in 2022, we had the first population suppression gene drive proof of concept demonstrated in lab mice in Paul Thomas's group at the University of Adelaide. This work used a naturally occurring selfish element, the T haplotype, to spread faulty copies of a female fertility gene down generations in the lab. So just briefly, the T haplotype, it's something only found in mice. And the way that it works, it's overrepresented in the sperm of male mice because it contains alleles that both impact the development of all the sperm in the gonads, but protects the sperm at the same time that carries the T haplotype against that impact. So because of this suppression effect and restoration effect, you find that the T haplotype and anything associated with the T haplotype is overrepresented in the sperm and the next generation of offspring. So the question then is, can we build on our understanding of the mechanisms that are working in mice for development and application of genetic control approaches for rats? The first stages here are that we've been supporting 3 candidate technology approaches, initial explorations to see can building on this understanding of this mechanism of the T haplotype from mice guide us to some usable advances in rats. The first stream of work that we've supported is through Genomics Aotearoa again, and it's being conducted by Ag Research or led by Ag Research. And they've been conducting separate trials in Norway rats, investigating different potential components of rat genetic control. The key thing here is it's not bringing those components together yet. Even though this work is in containment, very strict containment, we're not creating gene drive rats. We're just looking to see how well do different components of that T haplotype work if they're brought into transgenic rats. The first part of that is seeing if those components of the T complex that together make it a selfish element would be functional in a Norway rat. And the second part of this is testing the effect of a knockout of a female fertility gene on Norway rat breeding. This work is still ongoing this year, and we hope to see the results coming out from this in the second half of this year. The other 2 streams are both ongoing in the Paul Thomas group at the University of Adelaide. As I mentioned, Paul and his colleagues they did the initial 2022 demonstration proof of concept in mice of a population suppression gene drive. So this work here is actually going back into mice initially, but the whole idea is that can we artificially replicate that T haplotype selfish element functionality? If we can artificially replicate it in mice, we can do the same artificial replication in rats, as long as we're using mechanisms and components that occur in both species. For the fast drive, 2-step process again, similar to how the T haplotype works in mice. So the first step is, can we replicate that impact on the development of all the sperm in the gonads? And the second stage is, can we actually add then that T haplotype protection to the sperm carrying the artificial haplotype only? Some progress has been made here. The first stage has been passed very, very successfully. So we can do that first bit. And the second bit, again, should be completed by the end of this year. The third candidate approach that we're looking at, again at Adelaide, cleave and rescue is a gene drive approach that's been demonstrated in vertebrates. It's never been demonstrated in mammals before. So we're initially funding this work in mice. And the modeling suggests it could actually be a lot more effective for mammalian population suppression gene drives than other approaches such as homing drives. Again, there's two key stages we're exploring right now. Similar to the ag research work, is the cleavage of genes that are essential, in this case, for sperm fitness. And then can we create gRNA resistant versions of the sperm fitness gene that would then confer the rescue of sperm fitness to those cells that have the haplotype. In this situation, again, for this development, the first step has been passed very well, again, is that we've shown that we can effectively cleave the genes essential for sperm fitness. And now we're working on that second piece, the rescue of sperm fitness. So just to finish off, where will this go to next? Of course, the modeling work will continue, the social work, the cultural exploration, the international policy engagement. But in terms of the technical work, really, if any of these technology candidates are sufficiently successful from the 3 explorations going on now, the next stage will be to actually add female fertility impacting components to them to see actually can we get population suppression happening down generations in mice in containment, or in the rats being worked on at ag research. The second step then, of course, would be to actually show over several generations still in containment is, can we get this population suppression functionality working at a level that the models predict could cause effective control in the wild. So that's where we're at. We've got these 3 technical explorations ongoing. We hope to see the results from all of them by the end of this calendar year. And then how they turn out will guide what we do next on the technical front. Thank you to all of the co-funders for all of the different parts of this presentation, this package, this portfolio of work. And I'll invite any questions.
All right, I'll do the real clapping. Others can do the magical yellow-handed automatic clapping. All right, thanks, Dan. That was awesome. It was a whirlwind tour of the many, many, many things that you guys are doing. That was really cool to see the overview like that.